Thank you all for coming. Today I'm going to talk to you a little bit about the hearing research that I do at Boston University and how Mathematica helps me with that. So the application I'm interested in is related to the cochlear implant neuroprosthesis. And in the healthy auditory system, sound impinges on the outer ear, goes through the outer ear canal and vibrates the tympanic membrane. And this mechanical vibration is transmitted via the middle ear bones to the inner ear, where this sound-induced vibration induces fluid motion that in the inner ear, called the cochlea, that moves hair cells in the sensory cells of the cochlea. There are roughly 3,500 of these sensory cells. And these sensory cells ultimately electrically excite auditory nerve fibers that comprise the auditory nerve. There are roughly 35,000 auditory nerve fibers. And these sensory cells excite electrically, essentially, these auditory nerve fibers. And then these auditory nerve fibers produce electrochemical impulses that travel from the ear to the brain. And it's the timing of these electrochemical impulses that the brain interprets as sound. In the deafened case, the sensory cells that produce these electrochemical impulses in the auditory nerve are typically dead. And so these electrochemical impulses are not produced. And so there's nothing for the brain to interpret as sound. However, since about the late 70s, they've been able to restore a degree of useful hearing to these subjects by electrically exciting the auditory nerve. The way this device works is there's an external microphone that collects sound, and this microphone signal is wirelessly transmitted through the surface of the skin into an implanted electronic device that figures out how to stimulate a series of electrodes, and these electrodes artificially excite the auditory nerve fibers, causing them to produce these electrochemical impulses that the brain can interpret as sound. This is a diagram of the situation. This is one of the thousands of auditory nerve fibers, and normally this would be connected to a sensory cell, but in cases of deafness, those sensory cells are dead, typically, and the nerve ending degenerates. However, there's still a region that contains voltage-gated membrane proteins, voltage-gated ion channels that can produce this electrochemical spikes that propagate towards the brain, which the brain can interpret as sound. And this is the stimulating electrode that excites this spike generator with current pulses. And this is an example of an experimental record of what the voltage looks like at the spike generator as a function of time. And these little markers down here indicate the time of the stimulating current pulses. And the amplitude of these current pulses are the same uniformly. And this is the voltage versus time record. And you notice that there are these large excursions here. Those are the action potentials, or neural spikes. And it's these waveforms that propagate along the nerve. And it's the timing of these spikes that encode the information. One thing you notice about this record, although the input is completely uniform, they're uniformly spaced pulses, and they're all of equal amplitude. You notice the timing of these pulses is irregular, consistent with a chaotic response. And another feature that you notice from this plot is the so-called refractory feature of the neural spike generator. So here there was a spike in response to this pulse, but the subsequent response was very suppressed. This is the refractory behavior. It has to do with the fact that the channels that let in the ions are either inactivated in such a way as to suppress the next response. But this response eventually recovers with time, but there's a complicated interaction between the stimulating pulse and the recovery dynamics. And so what we want to do is understand this relationship between the input shock train that you put in and the output spikes. And that requires understanding what's going on at the spike generator. One way of approaching this is to model the biophysics of the situation, which are roughly captured by the Hodgkin-Huxley equations. But then there are simple differential equation models you can use of the Fitz-Unigumo type, which are related to the van der Pol equations. But what I'm really interested in, what I'm currently exploring, is whether it's possible to reduce this more radically to a one-dimensional map description that might allow you to get some geometrical insight into the properties of the spike generator that would also give you insight into this fundamental input-output relationship, which might, in principle, allow you to adjust your shock train to produce the best output for the person's hearing perception. So this is, I'm going to briefly talk about this biophysically based differential equation approach. Very briefly, I don't have time to go into the details. They teach whole courses on this topic, but here's kind of a circuit diagram of what goes on at the, in the biological membrane that defines the auditory nerve fiber. There's, there are charge-carrying species, sodium ions and potassium ions that can carry a sodium and a potassium current. That's represented by these quantities here, and there's a certain resistance they feel through going through the membrane, represented by these resistors and an associated conductance. Then there are equivalent potentials produced by the concentration gradients of these ions. And this is a representation of your current, your electrode that applies a current across the membrane, and this is the cross-membrane capacitance. This is a leakage path. So if you write down the differential equations, oh, I need to say one other thing. These variable resistors are critical to the spike generation process, and they're related to the intrinsic electrical properties of gating proteins that let in these charged ions. I'm going through this very fast, but I just want to give you a sense of this mathematical description. So there's these quantities M and H that are related to the open probabilities of protein subunits. That determines the conductance to the sodium ions, and there's another quantity N that determines the probability that the potassium channels are open. Those are important variables in the Hodgkin-Huxley model. So if you apply Kirchhoff's law and Ohm's law to that circuit, you get this differential equation in terms of G's and this M, H, and N factors. So that's one of the differential equations of the Hodgkin-Huxley set, but coupled to that are these three additional equations that tell you what these protein subunits are doing, And this is in, they are governed by classical rate laws that have voltage-dependent rate constants that are empirically derived. And so you have a fairly complicated differential equation set, but it does do a good job of reproducing this as an action potential like the empirical record. So there's a pulse here indicated by this circle, and you have this large excursion. But then also this refractory property is reflected in a reduced response here, but eventually recovers. And this is what the probability of all these protein subunits, what they're doing at the same time. But that's all pretty complicated. It would be a four-dimensional phase space. But it was discovered by Fitzhugh and other people that these much simpler differential equation sets behave qualitatively similar under pulsatile stimulation. And they only have two variables, one that's like the voltage variable, and another one that captures sort of all the refractory behavior. So when y is large, it suppresses x, so you get a small response to the second pulse, even though you got a nice spike to the first pulse. So the simpler system behaves in a realistic way, even though it doesn't incorporate the biophysics. And you see how nice this is. It's just 2 dimensions, 3 parameters, and it's completely linear except for this one cubic nonlinearity. But what's really amazing about this, and what I'm pursuing, is that when you start visualizing the solution in the phase plane, which you can do easily because it's only 2 dimensions and not 4 or higher, you see that the solutions follow these loops. So this is the resting state, and a pulse comes in, takes you across a quasi-separatrix, and you go through this long excursion. This is the action potential path that it follows, and these are the refractory responses. When you start looking at the problem in this way, you see that there's an even more radical simplification that may be possible. So, and this shows the solution to these Fitzhugh-Nagumo equations. Mathematica is in the background with ND-solve, solving this in real time. And this locator simply locates your initial condition in the phase plane of x and y. X is your voltage-like variable. And this point is near rest. And a pulse will displace you. And if the pulse is big enough, it displaces you across the separatrix. And you go through this long excursion. And then you can play around with, let's see, another thing is if you displace the pulse by the same amount, but y is bigger, then you don't get that large excursion and you run back to rest. That's the refractory property. And when you play around with this, you notice that there's this behavior where no matter where you start, you rapidly join this red path, which we call the home path. And so that kind of suggests that the problem, at least to first approximation, might actually be one-dimensional, at least to an approximation. And we actually exploit that. The trick is you mark off, you start your solution here on the home path, and you mark off equal moments in time. And so you notice here, these points are widely spaced because it's moving very rapidly when it's on the home path in this segment. But then as you get close to rest, things slow down. And so this defines an internal time. And so the whole trick is a pulse simply resets your internal time. And that's illustrated here. This allows us to control the location right before the pulse. So I'm dialing in to a large value of tau, which is near rest. So here is your location before the pulse. The pulse comes in, displaces you across the separatrix, and this is the path that ND-solve computes by solving the equations. This large black dot is where you are immediately after the pulse, and you can evolve this forward, and the black point rapidly joins up to the red path. But there's also this green dot, and this green dot is sort of the one-dimensional approximation. If you simply say, instead of solving the differential equations, if you just say, well, the effect of the pulse is to reset your value of tau to some small value, you get the right answer after a short period of time. So very quickly, as you evolve time, the blue point that you reset immediately back to here joins up with the black point. And so as long as you don't care about really short times, which you don't in this case, the problem is effectively one-dimensional. And you can consider other initial values of tau. For example, let's say your tau is not far enough advanced so you're really close to rest, and instead you're at a slightly earlier value of tau, but not too much earlier. You're sufficiently, Y is sufficiently big to suppress the large response, but you still get a little time resetting. You're moved back, instead of from around tau equal 20 all the way back to tau equal 4, you're moved from a large value of tau to a slightly smaller value of tau. Is at 10, tau equals 10. So a pulse that arrives when you're slightly more refractory sets you back a little bit in time, but not a lot. This shows the approximation and how the blue and the black join up. And then the other obvious case is what happens if a pulse comes in on top of an action potential. An action potential lives over here when you join this branch, so you can dial up some small value of tau. And here, you see our little black dot is obscured by the green dot. That's because the pulse has really no effect. You could ignore, the effect of resetting is 0 in this case. The pulse has no effect in this extreme case. And so you just reset it to exactly where you were right before the pulse. And then you see they join up. And so if you make a map for, if you ask, where are you reset to for every initial value of tau, then you've sort of captured the entire dynamics of the system for a pulse of that amplitude. So we showed before that any value of tau that's really close to rest will get reset to something like tau equal 4. That's this flat portion. So all these points that are really, really close to rest all get reset to around 4. The other easy case is this really small values of tau. The pulse has no effect. And so if you're at 5 and a pulse comes in, you're reset back to 5. And so you already have two features of the map. It has to be flat out here and linear slope 1 here, and they have to join up somewhere. This region here is that intermediate region where you get a little resetting. You're not set way back to 4, but you're still retarded slightly from, say, 14 to 13 or something like that. And the form of this map changes smoothly with the pulse amplitude. And this is work in progress. Amazingly enough, if you apply that same algorithm in the higher dimensional space, you get similar looking maps. So you have this one-dimensional geometric interpretation. A one-dimensional geometric representation of the response that even works for the complicated biophysical model. And two things, Andrew Moylan worked with me last summer, and there are a couple features that vastly improve the performance of this. They're really critical things that when I incorporated them, they sped up my algorithm by 800 times. One is event handling. So it's really important in this problem for the differential equation solution progress to be continuously monitored. So because you want to stop integration as soon as you join up to the home path, or else you're wasting computational time, like my old algorithm did. And the second really important thing is this function called nearest, which places the home path with this geometrical object in 2 or higher dimensional space into an optimized tree structure that allows for you to rapidly compute the distance between any point on this plane to the red curve. And it's not totally trivial because you need to find the closest point on the red curve. So in summary, for a wide range of pulsatile stimulation conditions, it appears, this is work in progress, that indeed even these complicated differential equations can be fairly well approximated by this one-dimensional map, and the hope is that that will provide some geometrical insight into this critical input-output relationship. And then there's certain advanced features of Mathematica that help out a lot in computing these maps. And I'd like to acknowledge Andrew for his ongoing assistance in developing and refining this algorithm. Thank you.